Hi everyone. Today our topic is Mendels Law. What is the Mendels Law? In detail we will study. Mostly we have a very important part is that Mendel's three important law. First one is the law of dominancy. Second one is the law of segregation, and third one is the last one is the law of independent assortment. These are the three important law is present in your syllabus. Now we will see one by one. What do you mean by law of dominance? Here is very simple. You know that a single character is expressed by a pair of factor. Means here Mendel give the name as a factor, but nowadays we call as the gene. We call as the gene. But before that, Mendel is known as the factor. Means what happened? A single character means this single character expressed by a pair of factors. Means it having the pair of factors. Here what happened? Example: the Pisum plant, P plant. Here they select a one character. Means we call it the tallness. Tall character will be selected. This tall character is selected, it having the pair of factors. Here we are representing the pair of factor with the help of the capital letter. If suppose it is a dominant, we are denoted as a capital T. If suppose it is a dwarf variety, they got this type of the small t. Here capital T means here this Mendel selected Pisum plant. Mostly it studied on the stem of that P plant. Here what happened? They are select one character. It having the two varieties in that plant, P plant. On the basis of their tallness, it having the two varieties. One is the tall and another one the dwarf. Here we know about that single character is expressed two pair of factors. If suppose it is a tall plant, it is showing the two factors, pair of two factors, one capital T. And if suppose it is a dwarf plant, it will be showing dwarf character. That time also express the pair of two factors, small t. Now, if suppose it is a tall plant, we know that we already studied the dominancy and recessive. Dominant means those are the dominant character will be expressed in next generation. That type of character with the dominancy. Recessive means they are suppressing the character in next generation because of this dominant gene. That type of this factor we call it the recessive factor. Here dominant factor, recessive factor. Now what do you mean by law of dominancy? Here law of dominancy is very simple. When this tall plant is crossed with the dwarf plant, it will be denoted by the dwarf plant, or we call it the recessive gene. A recessive factor we are denoted by the small letter small t. Here we are denoting here capital T. Now what happened? Then it is a parents. They are crossed with each other. These two plants is crossed with each other to form a F1 generation. We call as the first it will be form the gametes, capital T, and here form a gamete small t. Means two gametes will be formed here from the one tall plant, from the one dwarf plant. Means these two gametes are fused together to form a F1 generation. Means capital T, means small t. There we found this type of the tall plant, but it is a heterozygous type of the tall plant. Means what happened here very important, showing the law of dominancy. Why we are showing here law of dominancy? Because these are the nothing but the tall plant. Heterozygous condition, but it is a tall plant. We already studied what I mean by heterozygous. Heterozygous is nothing that the different alleles of a particular gene. Means we call it the heterozygous. But here what happened? This is a heterozygous condition, but here the phenotypically it is showing the tall plant. Why it is showing the tall plant? Because it contains dominant factor capital T. Means what happened? This dominant factor is mask the expression of the another factor. Means what happen here, these capital T showing two important functions. One function is that they expressing their characters, and another important role or functions of that capital dominant factor is that to suppressing the recessive factor, their expression. Their expression will be suppressed by the dominant factor. This is nothing but that, what happened? This is nothing but that we call it the law of dominancy. Simple. We will study about the law of segregation. It is also very important for the genetics. Because of this law of segregation, we know about how they will be segregated. It is very simple. Now we will take one example. Everyone knows that, we know about that a single character showing a pair of factor, or we call it the gene. But these characters of that organism present in bundles, means lot of characters are present in that organism. They are present in that organism in the bundle, but they are not contaminated to each other when they are present in that organism. When it will be present in that organism, lot of characters, we are expressing our character. Means we having the multiple characters. Means all character expressed by a pair of genes. And these all pair of genes or these all characters, they are present in bundle of our body or in that organism. What happened? It is not contaminated to each other in that condition. But when it will be formed the gametogenesis, means formation of that gamete, that time what happened? It will be contaminated. Means it will be what happened? It will be segregated. You know that gametogenesis is nothing but that formation of that gamete. When it will be formed gamete, when it will be formed the gamete, it is very simple. Gamete will be formed, and when diploid number is converted into the haploid number, means what happened? A reduction of that number will be occur here. Means what happened? Gametogenesis. When gametogenesis will be occur, that time what happened? These genes will be separated into the gametes. Means diploid is converted into the haploid. Means it is a 2n number. It is converted into the n number of chromosomes. Means here n number will be divided. Means there will be separation will be take place. Suppose we are taking the heterozygous tall plant. This is a heterozygous tall plant. This heterozygous tall plant, when it will be separate at the time of gamete formation, when it will be separating, that time what happen? Law of segregation will be occur here. Means those are the genes, they are present in that organism. They are not contaminated into each other, but they are expressing their character. But at the time of gametogenesis, these pair of factors or genes is separated in that separate gamete. We call that the law of segregation. Here what happened? This heterozygous tall plant, if suppose it will be formed the gametes, that time what happened? Here two gametes will be formed. First one is the capital T, another one the small t, means separation. Here segregation will be take place. This is nothing but the law of segregation. Now we will be see the law of segregation, law of independent assortment. Now it is also simple law done by the Mendel. But simple, you know, it is an example of the di-hybrid ratio. Means it having select two characters. On that basis, the law will be explained. This law of independent assortment means what happened at the time of gamete formation. That time what happened? These characters or these genes, they are separated, but they are separated and they are independently separated. means they are independently separated. Example, you know that if suppose in the B.Sc. second year she is coming on in class, but her friend is not came, means we call it the independent one. And if suppose her friend is also come with that girl, means what happened? They are dependent one. Means dependent means what happened? They are dependent. If suppose she is coming to college, that time her friend is also came to that college, means what happened? They are dependent one. And independent means what happened? This girl is came to college, but her friend is not came, means we call it the independent one. Or sometimes they came or not came, they are independent one. Either it is means what happened? We call it the independently, means they are enter into the next generation or in that gamete, they are independently. This is nothing but that we call it the law of independent assortment. Example, you know that yellow round. We are taking here example of the heterozygous yellow round plant, means with having two capitals. We are taking one is the heterozygous one, capital R, small r. It is a heterozygous yellow round plant. Here what happened? Independent assortment means what happened? Here this capital Y, when gamete formation, at the time of gamete formation only, this type of the independent assortment will be occur. If suppose this gamete formation, that time what happened? This capital Y is come either capital R or it come either small r, means what happened? This Y is independently. तुम्हाला यायचं असेल तर या, नाहीतर नका येऊ, पण मी जाणार आहे आणि माझ्यासोबत कोणी असेल तर त्याच्याशी मला घेणं देणं नाही. म्हणजेच काय? इंडिपेंडंट असॉर्टमेंट म्हणजे माझा तुमच्यावर अवलंबून नसणे. म्हणजे जेव्हा फर्टिलायझेशन म्हणजे झायगोट फॉर्मेशन किंवा गॅमेट फॉर्मेशन होत असतात त्याच वेळेस हे लॉ ऑफ इंडिपेंडंट असॉर्टमेंट बघायला भेटेल, आपल्याला दिसेल. हियर इज व्हेरी सिंपल. दिस Y is came with this capital R, this allele or factor, or it is also come with the recessive allele, or we call it the recessive factor, small r. Means this Y is independent one. This Y is independent one. For that purpose we call it the law of independent assortment, and it is an example of the law of independent assortment. Thank you very much. Subscribe my channel and share and like. Thank you very much.